So we're gonna skip to the next lecturer, Professor Nicola Simola. He's a neuropharmacologist and neuroscientist. He currently is an associate professor in the pharmacology of pharmacology in the University of Cagliari in Italy. His main research interests involve in neuropharmacology and neurotoxicology of psychostimulants and the neuropharmacology of dopaminomimetic drugs in experimental rodent models of addiction and neurodegenerative diseases. So, Professor Nicola, the floor is yours. Thank you very much.
Okay. Please let me know if you can see my screen and if you can hear me.
Yes.
Okay. So good day, everybody. First of all, I would like to thank the organizers for inviting me to this very interesting meeting. Today, I will present the results of a recently published study by our group that proposes 50 kHz ultrasonic vocalization as a marker of the affective properties of dopaminomimetic drugs in hemiparkinsonian rats. So Parkinson's disease is the prototype of movement disorders, and it stems from the degeneration of mesencephalic dopaminergic neurons that are located in the substantia nigra pars compacta, so in the deep brain, as you can see from this cartoon. And this degeneration of dopaminergic mesencephalic neurons results in a loss of dopamine in the extrapyramidal ways, leading to an imbalance in the function of striatal efferent pathways, eventually resulting in motor impairment. As this cartoon shows, so I will go very fast here, there are two different striatal efferent pathways that are regulated by dopamine, so the direct pathway and the indirect pathway. In physiological conditions, this pathway work in a balanced way, so they process motor signals and they return motor signals to the motor cortex, allowing the correct performance of movement. Where dopamine, mesencephalic dopamine, is lacking, there is an imbalanced functioning of these pathways with an aberrant processing of motor inputs, resulting in motor impairment. So the cardinal symptoms of Parkinson's disease are motor symptoms, for example, akinesia, bradykinesia, muscular rigidity, resting tremor. However, in recent years, it has increasingly emerged that non-motor symptoms as well are important in Parkinson's disease. These may be symptoms related to the disease itself, for example, cognitive decline, vegetative symptoms like hypotension, sleep disturbances. Many of them may also precede the disease, the onset of motor impairment. And there are also psychiatric disturbances. These can be idiopathic, for example, depression that may precede the onset of motor impairment, or iatrogenic, namely caused by the use of drugs that are employed to treat motor impairment in Parkinsonian patients. Indeed, Parkinson's disease is the only neurodegenerative disorder for which an effective therapy exists. With therapy, I mean a pharmacotherapy that is able to counteract the symptoms of the disease, but not the progression of the neurodegeneration. So for Parkinson's disease, there is the so-called dopamine replacement therapy that is made of the dopamine precursor levodopa, which may be associated with dopamine receptor agonists, like, for example, apomorphine, pramipexole, and others. And this therapy is very effective at counteracting motor impairment. However, as I was saying, this therapy does not arrest the progression of neurodegeneration, and it has also some untoward effects. For example, with the progression of the therapeutic program, there is a waning of therapeutic efficacy, the emergence of motor complications, especially with levodopa, so the so-called dyskinesias that are very limiting for the quality of life of patients, and then the iatrogenic psychiatric-like disturbances. These are a series of symptoms that include, for example, impulsive compulsive behaviors like binge eating, hypersexuality, kleptomania, psychotic-like manifestations that may involve, for example, hallucination, delusions, and are very similar in their manifestation to idiopathic psychosis, and the so-called dopamine dysregulation syndrome, that is an addictive-like behavior consisting in the, let's say, in the addictive-like use of dopamine-replacing medications, like, for example, levodopa. So as you can see from this cartoon taken from this article published in the Journal of the Neurological Sciences, these disorders can be manifested individually or in combination, and they may heavily limit the quality of life of patients. It is now very well known that there is a limited number of patients affected by these disturbances overall. As you can see from this cartoon, that the percentage of patients, Parkinsonian patients affected by behavioral disturbances like gambling, sex dysregulation, compulsive buying, compulsive eating, is modest even compared to the healthy population. However, the problem is that as of today, there is an unsatisfactory therapeutic management for these diseases, because there is a limited number of drugs, a very low number of drugs that are approved for treating this disease, and are usually drugs that are used off-label. And a reason why there is such a low number of drugs may be that there is a limited number of reliable animal models that can be used to study these disturbances at the preclinical level. And having adequate animal models may contribute to increase the knowledge about the mechanisms that underlie these disturbances, and about the mechanisms that can be targeted for treating these disturbances. So a possible origin of these iatrogenic psychiatric-like disturbances is a modification in the emotional state of the individual, of the Parkinsonian individual. And there are several studies performed in experimental animals and humans. There are some examples that demonstrate that drugs using dopamine replacement therapy may modify the emotional state in both experimental animals and humans. Therefore, it may be useful to characterize novel experimental tools that allow to investigate at the preclinical level the affective properties of drugs that are using dopamine replacement therapy. So these novel experimental tools could be useful to understand whether these drugs affect the emotional state and to what extent they do so. So ultrasonic vocalization, we focused our attention on ultrasonic vocalization, so I will go very fast here because Professor Vore has already provided a very extensive discussion about this. But as you can see from this cartoon, there are mainly two categories of ultrasonic vocalization. So the 22 kilohertz ultrasonic vocalization that are associated with negative emotional states, and the 50 kilohertz ultrasonic vocalization that are associated with positive emotional state. Among the latter category, there are the flat and the frequency modulated ultrasonic vocalization. The flat ultrasonic vocalization are thought to have mainly a social coordination function, whereas the frequency modulated are thought to be those more tightly linked to the presence of positive affect. So as we know from previous studies by our group and many other groups, ultrasonic vocalizations, 50 kilohertz ultrasonic vocalization are emitted in response to and anticipation of stimuli that possess positive affective valence. Emission of this course is considered to express the presence of a positive emotional state. And the link between 50 kilohertz calls and the presence of positive affect is substantiated by the evidence that these calls are stimulated by the activation of dopamine receptors in the nucleus accumbens, that is a crucial region for reward and the positive affect. And also we have collected in the past years evidence to show that numerous drugs that possess positive affective properties may initiate or modulate the emission of 50 kilohertz ultrasonic vocalization. This effect is particularly evident with amphetamine and with other dopamine-mimetic drugs like psychostimulants and dopamine receptor agonists like apomorphine. So having said this, 50 kilohertz ultrasonic vocalization can be considered as a relevant parameter to be evaluated in preclinical studies about the effects that the drugs elicit on the emotional state. We have demonstrated that measuring emission of ultrasonic vocalization in rats treated with psychoactive substances may allow to investigate the acute, long-term, and conditioned effects of these substances, in particular of those acting on dopamine transmission. Here is a study that was performed in our laboratory comparing the effects of amphetamine and apomorphine, two drugs that stimulate the dopamine receptors but have a different abuse liability, with amphetamine being the one with the higher abuse liability among the two drugs. And you can see that amphetamine induces sensitization in the emission of ultrasonic vocalization, whereas apomorphine does not. And animals treated with amphetamine emit ultrasonic vocalizations in response to the presentation of environmental cues that were previously paired with the drug. And this effect is conditioned because, as you can see from this graph here, only the animals that received amphetamine in the test cage emitted ultrasonic vocalization where they were present when they were presented the test cage again, whereas animals that were treated with amphetamine in the home cage, alternated with vehicle in the test cage, did not vocalize when they were re-exposed to the test cage in drug-free condition. So from this evidence, we concluded that measuring the emission of 50 kHz ultrasonic vocalization would be a useful approach to investigate the affective state of dopamine-denervated rats treated with dopaminomimetic drugs used in the pharmacotherapy of Parkinson's disease. And studying ultrasonic vocalization is also important, as Professor Wolf said, because many of the behavioral readouts that are classically used in animal models of brain disease may not recapitulate the whole array of modifications in the emotional state that feature a human disease, especially those readouts that are based on locomotor activity of animals, because there may be a divergent modification in locomotion and emission of ultrasonic vocalization. So to study the emission of ultrasonic vocalizations in hemi-Parkinsonian rats, we have adapted a protocol that we have already developed in our laboratory, consisting in the alternate administration of drugs in a test cage, followed by a withdrawal and a re-exposure to the test cage to test the long-term and conditioned effects of drugs. We focused our attention on 3 dopaminomimetic drugs that are used in the pharmacotherapy of Parkinson's disease: apomorphine, that is used in patients that present episodes of freezing; levodopa, that is the gold standard drug used in Parkinson's disease; and pramipexole, that is a dopamine D2 receptor agonist that is widely used. So this is the experimental protocol. Now we'll explain this step by step. What I want to say now is that we measured the total numbers of 50 kHz ultrasonic vocalization and the number of categorized calls, so flat, trill, and frequency modulated throughout the experiments. We did not see the emission of 22 kHz ultrasonic vocalization in any of the phases of the experiment. So first of all, animals were subjected to the unilateral dopaminergic denervation with 6-hydroxydopamine, which is the gold standard model used for studying the effects of drugs on motor activity in Parkinson's disease, or with sham surgery. And sham surgery involved the administration of saline solution instead of 6-hydroxydopamine. First of all, we let the animal to recover for 14 days. Then we evaluated motor asymmetry by using the Silent test. Animals were then subjected to habituation and then to pharmacological treatment on alternate days that were performed in the test cage or in the home cage. And in this phase of the experiment, we performed the evaluation of ultrasonic vocalization emissions, recalling behavior after acute and repeated administration. So vocalization were evaluated after each administration, although I am going to present only the results collected in day 1, 5, and 9 for the sake of conciseness, and also because the analysis was very laborious. And after a withdrawal in the home cage animals, this withdrawal was of 7 days, and the animals were first re-exposed to the test cage in drug-free condition to evaluate the emission of ultrasonic vocalization conditioned to the drug-paired environment, which lasted 10 minutes. and then they were challenged with another injection of the drug they previously received, so with an injection of either apomorphine, levodopa, or pramipexole, to test the long-term effects of this pharmacological treatment. So this is these are the results showing that the animals displayed motor asymmetry, so they had an unbalanced motor function and a marked depletion of dopaminergic neurons, and the level of the substantia nigra pars compacta. This effect was evident in all the groups, and in part, but significant, depletion of dopamine neurons was observed also in the ventral tegmental area, which is another nucleus of the mesencephalon that is adjacent to the substantia nigra. So the results, first of all, show that in animals treated with apomorphine, we observed that the drug was able to stimulate the emission of ultrasonic vocalization in both dopamine denervated and sham-operated rats, although the calling behavior was more marked in hemiparkinsonian rats, as you can see here. And when the animals were treated with a low dose of apomorphine tested, which was 2 milligrams per kilogram, sensitization of calling behavior occurred in hemiparkinsonian rats only. Whereas when the dose was increased to 4 milligrams per kilogram, calling behavior of comparable magnitude was observed in hemiparkinsonian and sham-operated rats. These results are consistent with our previous findings in neurologically intact rats, showing that apomorphine stimulates the emission of ultrasonic vocalization. The important finding here is that this effect is more marked and becomes sensitized in hemiparkinsonian rats. As for categorized cold, we saw that the lower dose of apomorphine elicited a more marked emission of categorized colds in hemiparkinsonian rats than in sham-operated rats here. The effects, however, depended on cold category and administration day considered. As for levodopa, we saw that levodopa stimulated calling behavior in hemiparkinsonian rats only. So this is a marked difference compared with apomorphine that was able to stimulate calling behavior in both groups of rats. And hemiparkinsonian rats again displayed sensitization in calling behavior in response to drug challenge administered after withdrawal. This is with a low dose. With a high dose, we observed calling behavior in hemiparkinsonian rats only, but no signs of sensitization, although there is a trend towards an increase. So here, the low dose is the only one that sensitization similar to apomorphine. As for categorized colds, we saw that either dose of levodopa elicited a significant emission of categorized colds in hemiparkinsonian rats, although the effect with the lower dose is observed only after challenge, and during the treatment there is a trend towards an increase, but these effects are not significant. And with the higher dose, we saw a more marked effect on the emission of categorized colds, and these effects depending on cold category and administration they consider. As for pramipexole, we were very surprised to see that this drug did not stimulate calling behavior in hemiparkinsonian and sham-operated rats at the low dose administered. At the high dose administered, the higher dose administered, so 4 milligrams per kilogram, there was a scarce calling behavior in hemiparkinsonian and sham-operated rats, and hemiparkinsonian rats displayed a more marked calling behavior than sham-operated rats on the first administration day only. But this effect was significant only compared to sham-operated rats and not to vehicle-treated rats. So the same effect was observed for categorized colds, so a very, very scarce emission of ultrasonic vocalization. And again, this difference between sham-operated and hemiparkinsonian animals after the administration of the higher dose of pramipexole tested, so 4 milligrams per kilogram, but only on the first day of treatment. So as for the re-exposure to the test cage, so after the treatment was completed, the animals were exposed to the test cage in drug-free condition and evaluated for 10 minutes. Animals treated with apomorphine displayed an increase in the total number of calls and in the number of frequency-modulated calls that were emitted in animals previously treated with either dose of apomorphine, and these effects were not observed in sham-operated rats. So the lesion makes apomorphine able to condition the animals to the environment where the drug was administered. The same effect was observed with levodopa. Again, here we see an increase in the total number of calls and an increase in the number of frequency modulated calls upon re-exposure to the test cage, although the latter effect was observed only in animals treated with a higher dose of levodopa tested. And again, it is important to see that this effect was not observed in sham-operated animals that were previously treated with levodopa. So again, the dopaminergic lesion makes levodopa able to condition the animals to the environment where the drug was administered. Similar to what was observed during the pharmacological treatment, we did not observe any increases in the emission of ultrasonic vocalization in animals treated with pramipexole when the animals were re-exposed to the test cage. So this experiment shows that there is a real conditioned nature of the calls that are emitted in animals treated with apomorphine or pramipexole when they are re-exposed to the test cage, because in this experiment we did the opposite. So we treated the animals with either drug, so two different groups of animals with apomorphine or levodopa in the home cage, alternated with vehicle in the test cage. So hemiparkinsonian rats that were treated with either apomorphine, we selected the dose of 2 mg per kilogram, or levodopa, we selected the dose of 12 mg per kilogram in the home cage, alternated with vehicle in the test cage, did not display an increased emission of 50 kHz ultrasonic vocalization when re-exposed to the test cage in drug-free conditions. So this indicates that the emission of ultrasonic vocalization in response to environmental cues previously associated with the pharmacological treatment is a real phenomenon of conditioning. And we also evaluated rotational behavior in these animals, and we saw that all the 3 drugs evaluated, so apomorphine, levodopa, and pramipexole, stimulated contralateral rotational behavior during repeated treatment and challenge. This is in agreement with the bulk of evidence that has been obtained by us and others in studies that evaluate the effects of anti-Parkinsonian drugs on motor behavior. And we also saw that only rats previously treated with apomorphine displayed contralateral rotational behavior when exposed to the test cage in drug-free condition. These results, again, is in line with studies with previous studies by others, and corroborates the hypothesis that the emission of ultrasonic vocalization on test cage re-exposure is a phenomenon reflecting drug conditioning, which is paralleled also by this rotational behavior. About rotational behavior, however, it is also important to observe that in the case of pramipexole, there is a divergent effect of the drug on motor behavior and vocal behavior, which is in line with the evidence demonstrating that the emission of ultrasonic vocalization is not simply a byproduct of locomotion, but is a phenomenon that reflects modifications in the emotional state. Here, we may suggest that the mechanisms of the hyperstimulation of dopamine receptors may be common to rotational behavior and ultrasonic vocalization, but they reflect, these 2 behaviors reflect different effects of the drugs tested on motor behavior and emotional behavior. So to summarize in a graphical abstract form, as you can see here, our results demonstrate that different dopaminomimetic drugs that are used in the management of Parkinson's disease elicit the similar effects on the emission of 50 kHz ultrasonic vocalization in hemiparkinsonian rats that are evaluated immediately after the drug administration or in response to environmental stimuli that were previously paired with a pharmacological treatment. Since the emission of 50 kHz ultrasonic vocalization is considered a marker of positive affect in rats, the present results may be useful for further characterizing at the preclinical level the effects that dopaminomimetic drugs used in the management of Parkinson's disease elicit on the emotional state. The results of the present study may have possible implications to the setup of novel experimental models to be used in the investigation of behaviors that reproduce in experimental rats the iatrogenic psychiatric life disturbances that are observed in Parkinsonian patients. Please keep in mind that this is a suggestion, so we are suggesting this based on our results, but we have to perform additional experiments to clarify which kind of psychiatric iatrogenic disturbances may be reflected by the emission of ultrasonic vocalizations in hemiparkinsonian rats. So please let me conclude by acknowledging the work of the persons who have collaborated in this study: Giulia Costa, Marcello Serra, Jacopo Marongiu, Michaela Morelli, and I thank you for your attention. And again, I thank the organizers for the kind invitation and for giving me the opportunity to share this data with you. Thank you very much.
Thank you very much, Professor Simola, for a wonderful talk. We do have one question from Professor Rainer Schwarting. I would like to, I will allow you to talk, okay? So you will be able to ask the question yourself.
Okay, can you hear me?
Yeah.
Yes.
How are you?
Yes.
Hi. Great talk. Thank you very much.
Thank you.
I have a number of questions, but I will limit it to one or two.
Okay.
And it's about the same issue, the relationships. So what is your thought? What are your thoughts about the relationships between turning and calling? For example, do the calls also start earlier with repeated apomorphine challenges?
Yeah, they do. We in the published study, there is a supplementary material about this question. So there is a detailed time course of the ultrasonic vocalization and turning behavior every 5 minutes. Here I have presented only the cumulative data for the sake of brevity. But with apomorphine, you see a very rapid increase in ultrasonic vocalization along with the treatment. So after, for example, the last administration of the repeated treatment, the animals vocalized immediately. But this may be an, how to say, maybe an effect that reflects also in part drug conditioning, in my view. Because when we do the repeated treatment, we just inject the animals and we put them in the test cage immediately. So we see a very rapid increase in the emission of ultrasonic vocalization, which in my view can be due to both the drug and the conditioning properties of the drug. Also, please keep in mind that apomorphine is has a very rapid onset of action. So it's not surprising that the drug gives rise to a very rapid increase in ultrasonic vocalization, at least in my view.
Thank you.
Okay. Okay.
Right. Thank you. Thank you very much. Are there any other questions to Professor Simola? If not,